So today for bio we'll be doing some revision for chapter 6 lah, because before that we already skipped to chapter 9 because of your exam. So now we move back to chapter 6. So this video will be I will brief about the chapter 6 lah, and then after that during class we'll do the workbook. Okay, so chapter 6 you have two subtopics. One is photosynthesis lah. The other one will be leaf structure. Basically, it's the adaptation for photosynthesis lah. Means how is the structure and then how does that structure actually helps in photosynthesis. So, definition of photosynthesis, you need to know also lah. Process, always write it must be a process by which plants synthesize. Synthesize means they produce lah, or you can say generate. But since it's photosynthesis, so it's better for you to write synthesize. Synthesize carbohydrates. These carbohydrates here should be glucose lah. Means C6H12O6 from raw materials using energy from light. Means they use light energy lah. Normally is sunlight lah. And then raw materials is carbon dioxide and water. So for photosynthesis you need to know how to write the word equation and the chemical equation. This one is the word equation, this one is chemical equation. So this one carbon dioxide plus water, and then up here light, down here chlorophyll, because if chlorophyll is not present no photosynthesis will be occurring. And then product is glucose and oxygen lah. Okay, for carbon dioxide they diffuse into a leaf through the stomata lah, because stomata is open and closed every day. And then water taken up by the roots and then transported to the leaf through the xylem. Glucose they used to make this one is like the food lor, and then they use this for respiration to release energy. Glucose also used in respiration lah. Okay, so this one is the word equation lor. So CO2H2O, and then C6H12O6 O2, and then to balance it add 6. Actually this one, the equation is basically the opposite of the respiration, aerobic respiration. So this one, if you can remember this equation you can also remember the respiration, aerobic respiration equation lah. Okay, chlorophyll, to define it it's the green pigment that is found in chloroplast. Okay, chlorophyll transfers energy from light into energy in chemicals for the synthesis of carbohydrates. So they change the light energy to chemical energy. For the synthesis of the carbohydrates, in this case is glucose lah. Okay, so we already know photosynthesis is they produce glucose and oxygen. So what is the use of the glucose? So the glucose will be transformed into, some of them will be transformed into starch to store energy lah. And then some will become cellulose to build cell walls, and then some they won't be transformed into other things. They will straight away used for respiration to provide energy. And then sucrose is for transport in phloem. Nectar is to attract insects for pollination, so that they can reproduce lah, the plants. Okay, after these three things the fourth thing you need to know is the importance of two types of ions. Nitrate ions, NO3 minus, magnesium ion, Mg2 plus. So Nitrate ions is for making amino acids lor. Means protein lah. But this one is the basic unit. Deficiency means not if the plant doesn't have enough nitrate ions, the whole plant will become stunted. This means that the whole plant they will have weak stems, and then the leaves become yellow lor, and dying leaves. Magnesium is for making chlorophyll. So they don't form the whole part of the chlorophyll, just some of the parts of the molecules. So the deficiency for magnesium ions will cause chlorosis, means the leaf will turn yellow from bottom of plants to the top . Okay, after these 4 things, these are the things. Limiting factors for photosynthesis. The last thing for 6.1. You have triol. This one you can relate it to chapter 5, enzyme, the limiting factors. Because photosynthesis is actually enzymatic activity . Means that they are controlled by enzyme. So the limiting factors is more or less the same to the enzyme . Temperature, concentration of carbon dioxide. If you relate to chapter 5, the factor for this case is the substrate concentration. Okay, so when temperature increase, photosynthesis rate will increase. Here . Because it's controlled by enzyme, when they reach a certain temperature, normally it's higher than usual . They will drop back and then drop very fast also. Because the enzyme begins to denature, so the rate will decrease very gradually. And then light intensity also . Because photosynthesis they need sunlight or any other light as long as there's light . So these 2 graphs are more or less the same . So they will keep increasing until a certain level. Other factors becomes the limiting factor, means that they are controlling the rate already, not the light intensity already, and then they will become constant . So other limiting factors is, for example, temperature, carbon dioxide concentration . And then carbon dioxide they will also have the same thing . When carbon dioxide concentration increase, rate of photosynthesis increase, but until other supplies in shortage. So for example, temperature is not high enough, light intensity is not high enough. So for this one, remember there's 3 limiting factors. Okay, so this is all for 6.1. So let's look at 6.2. This one's still not yet. This one is the experiment. This one is to investigate how carbon dioxide concentration, how light intensity, how the chlorophyll is needed for the process of photosynthesis. So let's see from carbon dioxide, the procedure. First step is to destarch the plant, means that we remove starch from the plant, and then after that we tie a clear bag containing sodium hydroxide, NaOH. This is to absorb the carbon dioxide from the surrounding . So this one around a leaf. So this leaf they don't have any carbon dioxide supply. So by right, according to theory, no carbon dioxide, no photosynthesis . And then another one one is NaOH, sodium hydroxide. The other one is water. So And after that, they place the plant with both of the leaf in bright light for several hours. And after that, test both leaf using iodine. Iodine test, if you read it back to chapter 4, it's the test for starch. So for observation, you will see the leaf from back containing sodium hydroxide will remain orange-brown because no photosynthesis occur. So no starch will be produced because no glucose. The leaf from control back means water should turn blue-black because they photosynthesis has take place, and then after that glucose is produced. Glucose is after that converted into starch. So let's look at the next one, the light. So this one, the procedure is more or less the same as the okay, so for this one, the procedure for light is actually more or less the same as chlorophyll. So for aquatic plants, we use the hydrogen carbonate indicator solution. So this one, they use this experiment for what? To determine the rate of photosynthesis for aquatic plants. But still the experiment setup can still be used for these 3 limiting factors especially for light. Okay. Procedure first step, destarch by placing them into dark cupboard for 24 hours. Means that they let the plant to use up the starch without doing photosynthesis to replace the starch. This is to ensure any starch present in leaves is used up and not affect results. Okay. After that, cover the leaf partially with aluminium foil. After you cover it, no light will be on the part of leaf already. So, and after that, place the plant in sunlight for a day, remove the leaf, and test for starch. Soon after that, you will notice that the part that is covered with the foil will remain because no photosynthesis occur because they didn't receive any sunlight. And then the other one, that is the other area that's exposed to sunlight, it returns to blue-black color. And then if they ask you to write the conclusion, this one will be the conclusion. Light is necessary for photosynthesis and production of starch. Okay, lastly, chlorophyll. Okay, drop a leaf into boiling water. This is to kill the cells and break down the cell membrane. And then after that, leave the leaf for 5 to 10 minutes in hot ethanol. Okay. They leave the leaf into the hot ethanol. This is to remove chlorophyll. So, color changes from iodine can be seen more clearly. Okay, because they don't want to do iodine test. Okay. Dip the leaf in boiling water to soften it. Okay. So this one, after they remove the chlorophyll already, they drop it into boiling water. And then after that, put it on the white towel, spread out the leaf, and then cover it with the iodine solution to test iodine for starch. And then after that, you will notice that green leaf, the entire leaf will turn blue-black because photosynthesis occur in all areas. And then after that, okay, this one, this is to test. Normally they will use this kind of variegated leaf means that one part is partially green, one part is green, the other one is the other color or white color. So this one you will notice that the area with green color means with chlorophyll will be blue-black because photosynthesis occurred. And then without chlorophyll means maybe white or yellow also can. They will remain as orange-brown because no photosynthesis occurs, so no starch should be stored Okay, this one plant cell constantly requires oxygen for cellular respiration. Okay, respiration and photosynthesis. For photosynthesis, they take in carbon dioxide and then release oxygen. Respiration, they take in oxygen, release carbon dioxide. But respiration, they do it whole day But for photosynthesis, they only occur when the light is present. So normally when sunlight is present, only there will be photosynthesis So at night, normally the plant won't do any photosynthesis. Okay, and then this photosynthesis they will occur in chloroplasts, which are found in the palisade mesophyll cells, spongy mesophyll cells, and guard cell. So mesophyll cell and guard cell. Mesophyll cell you have two types palisade and spongy. Okay, light is vital for photosynthesis. Suitable temperature are also the same. Okay, let's look at the other table. This one is what, Okay, this one is to test the concentration of carbon dioxide Lowest to highest, purple, magenta, red, orange, yellow. Atmospheric level is at the middle here Okay, this one when you test the hydrogen carbon indicator when you see yellow color or orange color means that more respiration than photosynthesis. Lower pH means the rate of photosynthesis is lower than respiration. If rate means that the rate of photosynthesis is the same as the rate of respiration. If it's magenta or purple color means that the rate of photosynthesis is higher than rate of respiration. So more photosynthesis occurred That's why they have higher pH, more alkaline. If less photosynthesis occurred lower pH, more acidic. Okay, this one normally when there's light it will turn no leaf, there's light, red. Leaf, there's leaf means photosynthesis occur and then this one will be purple because net intake of carbon dioxide means that more carbon dioxide is taken in compared to carbon dioxide release. This one means that more photosynthesis occurred And then if it's yellow leaf and dark means that there is net take of oxygen. So this one respiration occurs more than photosynthesis No leaf and then there's light, red color means that or you can say this one is the control or you can say that this one the rate of photosynthesis is equals to rate of respiration. Okay, then this one is the last subtopic for chapter 2 chapter 6 leaf structure. So this one for leaf structure most leaves have large surface area and thin. So this one when they ask for adaptation of the leaf you can say talk about these 2 points large surface area for what? This one is to increase the surface area for diffusion of carbon dioxide and light absorption for photosynthesis. So for example, they ask you to give two adaptation of the leaf. You can give these two lor, but then you have to write lor. The leaves have large surface area, 1 mark. And then after that, this is to increase blah 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 blah. The other mark. And then this one second point, second mark. Thin is to allows the carbon dioxide to diffuse quickly lah. Okay, and then this one is the parts of the leaf lah. And this one you see if there's only dash means that this one is the points lah, normal points. If there's equal means that you can straight away use them as adaptation. Okay, so this one is the leaf structure lah. This one, upper epidermis is up one lah, and then lower epidermis. Okay, upper epidermis one cell thick only, and then up here you got cuticle, wax cuticle lah. And then after that, below the upper epidermis, you have mesophyll cells. This one long shape one oblong shape one is palisade mesophyll. And after that, irregular shape one is spongy mesophyll. And after that, the lowest part you have lower epidermis. And then these two to have holes for the carbon dioxide to diffuse in and diffuse out one. This one is guard cell, and then air space. And then after that, this one, chloroplast is inside the mesophyll cells and guard cells. And then this one is fascicular bundle, means that you have xylem and phloem in there lah. Okay, so this one is, we think typical plant cell looks like that from chapter 2, but actually this one is just palisade mesophyll cells. Every type of plant cells is different, but normally we see palisade mesophyll cells as the normal plant cell. Okay, let's look at the parts first. Chloroplasts have chlorophyll to absorb light so that photosynthesis can take place. So for adaptation, you can just use these 2 points. Cuticle. Cuticle is basically a protective layer on top of the leaf. The function is to prevent water from evaporating. And then adaptation. Cuticle is thin and made of wax. This is to protect the leaf without blocking the sunlight, because sunlight is needed for photosynthesis ma. Okay, guard cell, absorb and lose water to open and close stomata. And then this is to allow carbon dioxide to diffuse in, oxygen to diffuse out for photosynthesis lah. Stomata opens during day, closed during night. Evaporation of water also takes place from here, found in higher concentration of the underside of leaf to reduce water loss. So the stomata is usually placed on the lower epidermis because if upper epidermis means that the water can be evaporated easily already. And then the other, it is also the site for gaseous exchange. So basically stomata allows carbon dioxide to diffuse in, oxygen to diffuse out. Okay, upper epidermis. So this one is the adaptation. They are thin and transparent. And then this is to allow light to enter palisade mesophyll layer underneath it for photosynthesis. To take place. Lower epidermis contains guard cells and stomata. Okay. Palisade mesophyll cells. Okay, this one is the adaptation, and then this one is the function. Column sheet cells, they are tightly packed with chloroplast lah. And then they're at the top of leaf. This is to maximize light absorption. Means that they will need many sunlight to be there so that the chlorophyll they can absorb it lah. Okay, spongy mesophyll cells contain internal air spaces. This is to increase the surface area to volume ratio for gas diffusion. So mainly they use it for carbon dioxide lah. Okay, air spaces is to allow the diffusion of carbon dioxide through the leaf, and then more air spaces, more surface area. Vascular bundle means that xylem plus phloem. This is to transport substances to and from the leaf. And then adaptation thick cell walls of the tissue in the bundles. This is to support the stem and leaf so that the stem can stand upright lor. Xylem transport water. This one is the function lah. Transport water into leaf for mesophyll cells for photosynthesis and also transpiration from stomata. For the phloem, transport sucrose and amino acid. Okay, so this is all for 6.2 and the whole chapter lah. So that's all for today.